So in this session, I'm going to discuss the drawings from the student that's been submitted and look at how to improve your drawing so that your drawing will accurately represent the micrograph. Either you are given a slide where you need to be able to use the microscope to focus the structures clearly to draw, or you'll be given micrograph to draw. In both ways, your drawing must be accurate. Okay, so let us look at some examples and learn from the drawings. Okay, so here we have got the artery wall. So yeah, you're asked to draw a plan drawing of the artery wall. So this drawing, you can see that it's quite accurate, especially this region here. This region here is actually showing how the IEL are actually arranged in this artery. So I would say that this drawing is quite accurate. Can you see? The student is drawing here. You can even find exactly where is the location where the student is drawing that structure. Where is the curve? So it's quite a good drawing. I'll say that the thickness of IEL is also very near to the actual one. Okay, so the thickness of muscular layer, so the muscular layer is here, from here to here, all the way down up to here, because this is also smooth muscle. The only thing is that the orientation of the cell could be slightly different from the one over here. Okay. This one could be a longitudinal section. This one could be oblique section of the cell, like how you cut the carrot. So you look different. But both are smooth muscle, so it's considered as the same layer up to here. Then this is connective tissue. I think a lot of collagen fiber and elastic fiber here. Okay, so you can see that the drawing is accurate. The thickness is also accurate. So the marking point given will be first mark. Remember the format. Format of your drawing means we're looking at your pencil line. Pencil line is thin, sharp, clear, HB pencil, and the size of your drawing must occupy more than 3/4. So they may say, okay, the entire thickness must be more than 4 cm. So we'll measure. If it's less than 4 cm, you don't get the mark. So this is format. So format wise in this is quite good. Then the next one is your accuracy. Number 2 is accuracy. And then number 3 is label, if you're asked to label. All right. So accuracy, I can see is very accurate because the thickness of muscular layer is about 3 times of the connective tissue. So this part here is 3 times of this, is very good. Now, is the labeling correct? Lumen, good. Endothelium, no need to write tissue because endothelium is name of tissue already. Muscle is also tissue, so no need to write tissue, muscle, can already. Okay. Collagen cannot, because collagen, remember, is triple helix, is actually the molecule. So you're pointing to a tissue, you cannot label it as a molecule name. Collagen is a molecule name. So you need to label this as probably connective tissue, which contain maybe probably you have got the collagen, okay, tissue, and also maybe elastic tissue. Okay. So this drawing is quite good. Now I'm going to illustrate another drawing here. Now tell me, if you are marking the paper, how are you going to give mark for this student? Number 1, endothelium is only 1 single line and the student is drawing 2 lines. And it's telling me the endothelium is that thick, so it's totally out. Number 2, the student is not drawing the accurate drawing from here. I can't see anywhere here that the wavy line, it looks like this one. I can't find anywhere here the wavy lines like this. So it's not accurate drawing. Out, no mark already. Then number 3, smooth muscle and collagen tissue. How come the ratio is not correct? It's supposed to be 3 to 1. This is 1 to 1. So no mark. And then suddenly got this wavy structure here that did not appear in the micrograph. So what is this wavy structure? You learn in theory there's IEL and EL, but in different slides, different specimen, you may or may not find it. So you cannot simply draw. So this one, for me, I will only format out already. Accuracy out already. Probably I give mark for endothelium label, also wrong, right? Endothelium is this one. Okay. But I already give mark for accuracy because you got too many lines already in your drawing. So the accuracy drawing could be, there will be maximum 1, 2, 3, 4 lines, 4 horizontal line. And then the thickness of smooth muscle to collagen is 3 to 1. So accuracy, out. Labeling, endothelium, okay. Ignore the mistake here. Smooth muscle, okay. Collagen tissue, okay. So labeling 1 mark. So out of 3 marks, 1 mark. This is another drawing. Also quite accurate representation. You can see that the way the IEL curve, you can find it here. Okay, but not very accurate, but it's as close as possible. I think the student is trying to draw as close as possible, still acceptable. All right. Thickness wise, you can see the accuracy is there, 3:1. Okay, the only thing is the label is not correct because you need to label the name of the tissue, not the name of the layer. Remember, when I teach you this topic, I keep on stressing tunica, intima, medial, externa is name of the layer. So when you draw plan drawing, you're drawing the tissue. So when you draw the tissue, you have to label tissue name, not layer name. So no mark for labeling. And then vasa vasorum, actually, I am not sure whether this is vasa vasorum. I'm not sure. So therefore, I will not commit unless I can zoom into higher power to check, or else I cannot commit whether it's vasa vasorum here. Okay, so will I penalize or will I give mark? It depends. Because now I cannot see whether correct or wrong. Okay. Yeah, I cannot tell whether it's correct or wrong. So I just ignore this part. If I can see very clearly this is not vasa vasorum, I will not give mark for your drawing because it's not there. So you can only draw if you're very sure that that is a correct structure, especially when you want to label it. The moment you label wrongly, the rest of label correct, one label wrong, you get 0 mark for your label. Okay. This one, you can see that the accuracy, the wavy structure is okay, but it's too many lines. The epithelium cannot be a separate layer because it's very thin. You see not? You only see nucleus, you don't even see cytoplasm. Cytoplasm is as thin as one single line. So therefore, you can only use one single line to represent epithelium. So cannot draw this extra line outside there, right? IEL, good. The labeling wise, I don't like the tunica media is not correct. Tunica externa is not correct. So are you asking me to choose between these two, which one is correct? Every time in an exam, if I can see there are 2 answers and 1 correct, 1 wrong, overall wrong. No need to think already. Because you're giving the answer for the examiner to choose. That means you're not sure. If you're not sure, how can I give you mark? So you cannot write tunica media and tunica externa because you need to give name of the tissue. Connective tissue is correct. Smooth muscle tissue is correct, or smooth muscle alone is also correct because muscle is tissue. Okay. And then title, if you want to put title to your drawing, all title must be at the bottom of the drawing. Okay, why? Because if you hang a piece of picture, if you hang a picture on the wall and you hang it quite high, and then you have the name of the artist who paint the picture and the year that they paint the picture. If you put the title on top of the painting, you cannot read it because it's too high up. So you have to put the title at the bottom of the drawing, just like here, title is at the bottom of the drawing. So all title must be written at the bottom of drawing. And then you need to write figure number. If you want to write title, you have to write figure number. Like figure 1 is the first drawing for this exercise. So it's figure 1, artery wall, bracket TS. Then your second drawing is figure 2 dash whatever. Okay. For title, you need to write title for all the drawings that you do in the lab, so that in the future you want to refer back, you know what specimen you're drawing it from. But in exam time, you don't have to write title because you don't know what slide you are drawing. They may label it as L1, K1, M2, and so on. So you don't have to write title in the exam time. But for normal practical, you need to. This is for future references. Okay. Now, can you tell me when my micrograph is slanting to the left side, right? Why this student drawing is slanting to the right side one? How to give mark like that? You are reporting what you see, you know. People slant to the left side. Why are you going to draw people slant to the right side? Okay, so you want the examiner to minus mark, is it? Second thing, drawing is not accurate. Can you see the size of the lumen versus the wall? You look at this lumen is not long enough. You see this lumen is so long. This lumen is so short. And then the wall thickness is also incorrect. How come there are 3 layers in the wall? And the wall only got 2 layers. First is this layer here. Second is this layer. Finish already, right? The layer facing the lumen is the endothelium. Usually it's one line, one single line, right? Accuracy totally out. I won't give mark. Because why? People only got 1, 2, 3 lines. This student got 4 lines. And then the lumen so long and big, the lumen becomes very small. And then the wall is very thin, suddenly the wall becomes very thick. So accuracy mark out already. Format, okay. Format, you can get 1 mark. Okay, because pencil line is clean and clear. Same darkness throughout using 2B pencil. Right? And the size of the drawing is big. So that is 1 mark for format. Labeling wise, endothelium, smooth muscle, collagen tissue. Yes, so total 2 marks out of 3. This drawing, okay, quite good. Why? Because all tissues that can be seen are being drawn, including the blood. Don't forget, blood is made of cells, right? So something made of cells is tissue, isn't it? So therefore, you need to draw the blood that you can see here. See or not? So blood and label blood. Very good. And then you see here, these are fat cells. So it's adipose tissue. So draw and label adipose tissue. So this drawing is quite good, as in all the possible tissue you can see there, the student already draw and label. And quite accurate represent. Is it accurate? I think okay lah, acceptable. Actually, this layer a bit thick already compared to the wall. Right? But quite close to accuracy lah. You can see it. Overall, this one should be a full mark. This answer is full mark. Can see the student even label the lumen. So a lot of students forget to label lumen. Lumen is also one of the structure of the artery, so you need to label lumen. So I would say that among all the sample here, this is the one that will score full marks. Okay. This one, I would say that this drawing is more accurate than the previous drawing. Can you see that? This is the best drawing. This is drawing same standard as Miss Ho's drawing. Okay, very, very accurate. Okay, this one is very good. This is the best. Okay, but why the student go and draw individual cell? Are you drawing plan drawing or detailed drawing? Plan drawing, you outline the tissue. You cannot draw cell. If you draw cell, regardless whether it's big or small, when it's plan drawing, you cannot draw cell. The moment you draw cell, you lose 1 mark in the exam already. You see that? You see the fat cells are very big, right? But you cannot draw the cells. So that's the mistake in this drawing, right? Tunica intima, use 1 line to represent that. Tunica intima, sorry. Yeah, use 1 line to represent tunica intima, okay? Not 2 lines. Unless you can see it's 2 lines. Is it 2 lines? I can't see. 2 lines. 1 line. Only the part with nucleus I can see, the part with cytoplasm I cannot see. So therefore it's only 1 line. Okay, 1 line. You see, it's so accurate until it actually shows this part here, which is actually this part. Okay, this part rather than this part. Right, labeling wise, so accuracy, no mark, even though it's very accurate because you draw the cell. Okay, or maybe format wise, you don't get the mark because you didn't draw plan drawing. Okay, and then accuracy wise, maybe you get the mark, but actually this layer shouldn't be drawn. Then labeling wise also no mark because it's tunica intima media externa. So do you get a mark? Probably 0 mark. For such a beautiful drawing, such an accurate drawing, you get 0 mark, you know. My God, how can? And then you see this one didn't draw the blood. Okay, other than that, I think this drawing is quite good. Okay. So can you see that your drawing is very good, but then you may score 0 mark because of certain reason that I've explained to you. Okay, right. This drawing, also same thing lah. Epithelium is not a separate layer; it's only single line. So majority of students got the same mistake. Okay, When you have to draw it accurately, how can you draw a 2 lines for this one? You see it. You can't even see the cytoplasm. So why are you drawing 2 lines to show the thickness of the cell? But the student actually draw blood, so it's good. Endothelium, smooth muscle, collagen. But there's no adipose tissue drawn here. Okay, so probably you'll get 2 marks for one for format, one for labeling lah, for this one. All right, for this longitudinal section of the vein, I actually ask you to draw the vein, right? Ask you to draw the vein, not the wall. So here, this one is not correct because you didn't actually draw everything of the wall, because the wall have got this outermost layer is the endothelium, and then this layer is the, where's my smooth muscle? This layer here is smooth muscle, and then this layer is the connective tissue. Okay, connective tissue. So, and then here is the blood. So you need to draw the blood in the lumen. So this student got draw the blood, the lumen, up to the muscle tissue. But the muscle tissue got 2 types. You know, you got the muscle and you got the connective tissue. The connective tissue is missing lah. So when you got number of lines, then you don't get marked for number of lines, correct? Labeling, you get marked because labeling is correct. Format of drawing, you might get the mark because of the size, the pencil line, everything is okay. So maximum 2 marks for this drawing. This one is totally, this one is not answering the question. I asked you to draw the longitudinal section of the vein. So you need to draw the entire wall of the vein. So this one didn't draw entire wall. So it's already wrong. So total number of lines out already. Okay. And because you didn't label lumen and blood, because you didn't draw the labeling, I will not give mark. Incomplete labeling. So maybe format, I'll give 1 mark for correct, clear pencil line. Okay, big. So maybe 1 mark for the drawing format. This drawing, I like it very much because you can see that it's quite accurately drawn. All right. Again, endothelium is 2 lines. Really got 2 lines to draw endothelium. You can see. Yeah, maybe can. Probably in this slide, in this particular slide, there's a darker pink color here, a darker pink color here. Okay, which form the wall of the endothelium. So in this particular slide, because this darker pink is actually stretched throughout the entire endothelium, so probably you can draw this. Okay, 2 lines to represent the thickness of the endothelium for this particular slide only. Okay, then smooth muscle. Okay, connective tissue. That means the student is taking this as the smooth muscle, and from here onward, which is wavy, is connective tissue is okay. Because the student is using the texture and color to distinguish different tissue layer. I think this is quite okay. All right. Labeling wise, you need to use ruler to draw label line. If like this, you will lose 1 mark for labeling. Whether all your labeling is correct because you didn't use ruler to draw this line, I will give 0 mark for your labeling, regardless of whether all of them are correct. In this case, all of them are correct. So please make sure you use ruler to draw your label line. Okay. This drawing is very accurate because do you notice these 2 structures that the student draw? The student is actually drawing, reporting what he has seen. You see here, this is empty space. This empty space the student is drawing. So this one must be another empty space somewhere here. So this drawing is quite accurate, except for tunica intima, a bit too thick already. All right. And then please draw the blood inside the lumen. Please label the lumen and the blood. Okay. Title is good. Wall of vein. Title is good. All right. So this is for artery and vein. Now we want to look at the another